Hi students, now today we are going to study about translation in protein synthesis. See, now we are going to study translation means actually in protein synthesis, no, protein synthesis or the production of protein is completed in two steps. First step is transcription, which we have already seen in last video. Now we are going to study translation. See, what is translation? It is the second step of protein synthesis. First step is transcription and second step is translation. What happens here? Here mRNA results into the formation of protein. From mRNA, protein is synthesized over here. In transcription, what we have studied? In transcription, we have studied from DNA, mRNA is formed. Okay, and in translation, we will study from mRNA how protein is going to synthesize or formed. This translation is completed in three steps. I, E, T. I for initiation means starting. Second step is elongation and last step is termination. So let's start with the new topic, that is translation. What happens? We have already studied in transcription, mRNA synthesis took place inside the nucleus and this mRNA comes out through the nuclear pore into the cytoplasm. See what happened as soon as mRNA enters the cytoplasm from the nucleus. As soon as it enters the cytoplasm, all the amino acids which are present in the cytoplasm gets activated. Okay, activated means actually what happens over there, all the amino acids get attached to the specific tRNA and they form amino acid tRNA complex. I'll explain you with the help of one example. See this, can you see this? This is tRNA. Okay, what happens as soon as mRNA enters inside the cytoplasm, this tRNA or this amino acid becomes activated and they get attached to the end to this end of the tRNA, and this whole complex is called as amino acid tRNA complex. Now what happens as soon as this mRNA takes entry here in the cytoplasm? What happens here? Immediately the smaller unit of ribosome. See, ribosome is having two units. One is smaller unit, another one is larger unit. Always they are in the separated form. Okay, but as soon as there is the entry of this mRNA, this smaller unit comes and attached to the first codon. Okay, here just see the 30S ribosomal subunit, that is the smaller unit of ribosome, comes and attached to the first codon, that is start codon. Okay, and simultaneously when this ribosome is coming and getting attached, the first tRNA means here the first codon is AUG, means out of many tRNAs or out of many tRNA complex, the one who is complementary. Complementary means suppose here it is A is complementary to U. Okay, if U is here, A is the anticodon. If G is the nucleotide, here anticodon will be C. So here if codon is AUG, means opposite of this anticodon will be UAC. So they get activated and they come along with the ribosome. They come here from the cytoplasm and forms this type of complex. So it is visible to you here. This 5' end is the start end. This AUG is the first codon, start codon. Here what happens as soon as mRNA takes entry here, this smaller unit of ribosome get attached to the first codon and simultaneously this anticodon tRNA or the tRNA containing anticodon comes and get attached or forms bonding with this codon, first codon. See, bonding takes place. Now what happens when the bonding is taking place, the larger unit of ribosome also comes and it get attached on the smaller unit of ribosome. Now this is the 30S ribosomal subunit and this is the 50S ribosomal subunit. Now what happens? Codon anticodon bonding takes place here. Okay, And what happens? This ribosome is having three sites actually. One is called as E site, that is exit site. Next one is P site, that is called as peptidyl site, and last one is A site, that is amino acid site. Now see what happens when this bonding is taking place. What happens here? Bonding took place means it will release its amino acid. As soon as the bonding takes place between UA, CG, this amino acid is released. It is left over here, okay, at the P site only. Simultaneously, when this is taking place, what happens? See, the next codon is CCG. This is the next codon. So the tRNA which is complementary to this means, see, C complementary, or it is C is the codon, CCG is the codon, its anticodon will be naturally GCC. So this tRNA which is having the complementary anticodon will take entry through A site. Only first codon or only first tRNA get attached towards the P site, but rest all the tRNAs will come and attach to the A site only. Okay, this you have to keep in mind. First tRNA only will come and get attached to the P site. It is always AUG. Its anticodon will be always UAC. This is not definite, but the first codon anticodon will be definite. It is fixed. And what happens? As soon as the coding or the bonding takes place between codon and anticodon, this tRNA releases methionine. In eukaryotes it is methionine, and in prokaryotes it is F-methionine. Okay, it releases it methionine here. Simultaneously, when this step is taking place, another anticodon or another tRNA with anticodon comes and get attached to the A site. See, it gets attached with the, and it is carrying another amino acid. Now what happens? This bonding, once the bonding is over, this tRNA releases this amino acid, that is methionine, and it will move from, in this way, the remaining, or you can say the ghost tRNA, or the tRNA, or you can say empty tRNA will take entry or will get exit, it gets exit through the E site. It releases its methionine or amino acid and it exit from this side. What happens now? From this is A site, no? So this tRNA gets shifted here, or it get forwarded here. Okay, in this way what happens? The remaining one, the tRNA which leaves its methionine, or it releases its amino acid, get releases from the E site. Then what happens? See how it is releasing. This empty tRNA get released, and it is going again in the cytoplasm in search of another amino acid. Then what happens? See, another tRNA with third. See, this is the first codon, this is the second codon. Okay, as soon as the amino acid is left, no, this ribosome is also moving step ahead. This is first one, this CCG is second codon. Now the ribosome will move towards the third codon, that is UAC. Naturally, as soon as it reaches this third codon, the third tRNA with anticodon comes and again get attached towards the A site. Okay, it get attached. Again same process is there. This tRNA, after bonding gets completed, this tRNA leaves its amino acid and will get released from this side. In this way, continuously this process takes place. Okay, so this step is called as elongation. Initiation means where only there is entry of ribosome and tRNA, that is the initiation step. Now here what is happening? Here just the elongation of amino acid or polypeptide chain is taking place. Continuously the amino acids are coming and they are forming the chain. Okay, so here what happens? This is elongation where the complete polypeptide chain is taking place, and simultaneously ribosome is also moving ahead. But what happens when the ribosome moves towards the last codon? it may be UAA, it may be UAG, it may be UGA. When it reaches to the last codon, or you can say stop codon, or you can say nonsense codon or termination codon, what happens? This, for this no tRNA is there, because this, for this codon no anticodon is present. So what happens? Naturally the process of translation get breaks. Okay, and the chain, polypeptide chain formation also breaks. And what happens here, as soon as the process of translation stops, both the units of ribosome, that is the smaller unit and the larger unit, again get separated. And what happens, as soon as the ribosome gets separated, no, this mRNA, okay, now this mRNA work is also over. It releases freely in the cytoplasm, and immediately after the completion of this process of translation, it get denaturated by the enzyme nuclease. Okay, and hence we are always using the word mRNA is short lived, because their work is only to give the message as soon as there is formation of this polypeptide chain, or you can say as soon as there is a formation of protein, the ribosome, both the units of ribosome get separated and mRNA get denaturated. Hence this mRNA is short lived. So here we have completed translation process.